So is my screen visible? Mm,
Yes, sir.
okay, fine. So in today's class, we'll discuss the topics which were covered in week 9. That is characterization of isolated product. So earlier we have studied that how we can isolate a protein and how we can purify our protein of interest from the group of proteins. Now, once we have purified our product, we have our product with the help of different chromatographic techniques that we have discussed in our earlier classes. We have isolated and purified our product. Now we want to characterize it. Characterize it as in now we want to know what is the weight of our molecular weight of our product, how many monomeric units are present, whether the units are a heteromer or it is a homomer. So, and many other things. So, we want to characterize our product. We want to have the full knowledge of our product. So, for characterization, there are many methods which are used. So, as we studied in the first lecture, that we have to characterize our product. Once we have isolated our product, now we want to characterize it. So, characterization can be done on many levels and on the basis of many different properties. So, the first property would be your molecular weight, or on the basis of functional groups that are present in your protein and the sequence of the protein. So, then what we studied in this lecture was that for the characterization of molecular weight, we have many different approaches like gel filtration chromatography, electrophoresis, mass spectrometry. We have gel filtration chromatography, we have already studied in our previous classes. So, this whole week was particularly about electrophoresis and, in particular, SDS electrophoresis, because here we are dealing about product characterization. So, here we are dealing mainly with proteins. So, we have dealt not with the agrostia electrophoresis, which is used for the separation of nucleic acids, but for SDS, that is for proteins. Then, for the functional group characterization, we can have many spectroscopy methods that we might be studying in our subsequent lectures. And then the sequence. What is the sequence of your proteins and how the different amino acids are arranged and how the different monomers are arranged. So these informations will tell us about our product. Just a minute. Yeah. So these three information will tell us about the product. So gel filtration chromatography, we have already studied how to determine the molecular weight, that the radius of gyration, which is proportional to the molecular weight, and then next that we will be dealing mainly in this class is electrophoresis. So basic idea of electrophoresis is that your product, your protein, whatever your mixture of protein, whatever is there. So basically, any, not just protein, let's just say that any particle always experiences two kinds of forces. Okay, suppose if I have a particle which I am giving a force, so that is one force that I am providing. Okay, and always there will be a force which is opposite to the force that I am giving it, and that is the frictional force. So frictional force is always in the direction of opposite direction of that, of the motion and the force that is applied. So, and because of this frictional force only, there are a lot of things that we are able to do. Just like it's very basic. Like because of this frictional force, we are able to write. Because of this frictional force, we are able to walk. Because our shoes has grips, and because of that force of friction only we are able to walk. Because of the force of friction we can write on the blackboard and chalk. So in general what I'm trying to say is that if the force that is applied, it tends to keep the object in motion, or it tends to keep the object in stationary, frictional force will have different or just opposite effect. So just like frictional force exists everywhere in nature, so when a charged molecule or when a molecule is moving in an electric field, not a charged molecule, but I mean, yeah, it might be charged also if it has some like in SDS space it is charged also. Anyway, so when a molecule is moving in an external electric field, then there is a force which is responsible for giving its mobility, which is responsible for giving its electrophoretic mobility. And that force is this, your net charge into electric field. So this force is responsible for moving of the charge. So if this moving of your charge or moving of your particle. So your charged particle is moving. This is the direction of electric field. It is moving because of this force. And there would be a force which opposes the motion of this charged particle, which is your frictional force. Okay. And so this frictional force is causing this charged particle to resist the motion, to resist its flow. So why we have to study about this frictional force and this force which is responsible for its mobility is because, as we can see here, this is all frictional force. What this is all the formula, relative mobility. Okay. Because a charge, you see, if this is your gel, so these are the bands, whatever bands you get. So any charged molecule, it will stop at a point only where your frictional force is equal to the force that has been applied. Okay. And that point where the molecule, the charged particle is stopping, there you will have a band also. Okay. Where the frictional force is equals to the force which is being applied. This is like all the very basics of electrophoresis, and these mathematical small mathematical derivations and formulas, if you want to get into it, you can go also. They are not like very important. I mean, they could have been, I don't know there are some questions or not. I don't think that in some competitive exams they really ask questions related to these numericals. Maybe one or two, not like very difficult questions. So this is okay. You can just go through it for your knowledge and concept building. This is just like a principle of electrophoresis to get you know how electrophoresis work. And then we come to moving bound electrophoresis. Now, so what happens is that, so we know what is chromatography. So in chromatography, we wanted to separate our protein from the mixture of proteins that we have, right? So for that, we mixed our mixture of protein in some mobile phase, in some buffer. And then as that buffer was moving, our protein of interest was also moving through the column. And then depending upon which kind of chromatographic technique we are using, it will stick to the matrix or it will go through the matrix. It depends upon what kind of electrophoresis, chromatography we are using, whether we are using ion exchange or affinity or gel filtration. So that was the main objective. That means our protein, our mixture of protein was mixed in the buffer. Now, so for more advancement of this, so what they did was, what scientists this was that, okay, chalo, till now we were just moving the solution in buffer as it is. Now, or with the help of some pressure like HPLC. In HPLC, you have some more pressure. I don't think we have discussed up till now. We'll see if it is there in the course or not in the further subsequent classes. So what they did was that chalo, now we'll mix here. What we'll do, that we will dissolve the sample in our buffer only, but now we will also put it under electric field. So what will happen, that when we are putting our sample in under electric field which is already mixed in buffer, so the positive and the negative charged particles will separate. So now we will have distinguish that okay, in our protein these many are positively charged particles and these many are negatively charged particles. So it was like an advancement of the already existing technique, that here also the your sample is mixed in buffer only, but now we are providing electric field also. So this was the starting of electrophoresis. So in the starting of electrophoresis, so nowadays what you have a gel, right? You have a gel and you cast your gel and then you load your samples onto your gel. So you have a solid matrix on which you pour your sample. But when this electrophoresis technique started, then there was no solid matrix on which the sample was loaded. The sample was just mixed in the buffer, and then you then negative electric field was applied, and then samples used to your protein used to migrate. So that is why the name suggests moving boundary electrophoresis. That means there is no solid support, there is no solid matrix onto which your gel is loaded. So it is your moving boundary. This was initially it was really used. Nowadays nobody really uses moving boundary electrophoresis. Everybody uses the casting gel to load the sample. So the electrophoresis is carried in solution without a supporting media. So no supporting media. Supporting media means no gel was used here. So the sample is dissolved in the buffer and molecules move to their respective counter charges. Then sample is loaded in the middle of the U tube. You can see here. Here the sample was loaded, and then what happens is that charged molecules, it will move according to their charged, whether it will move to the cathode if they are positively charged or anode if they are negatively charged. So this was the first kind of electrophoresis, moving boundary electrophoresis. But then as scientists started doing experiments, so they observed that this kind of electrophoresis has certain disadvantages. Because what is happening, because your sample is mixed in the buffer, so separation is not very good, and there is overlapping of the samples also, because there are no separate lanes. Now what we have, now we have a gel, we have wells, and we have different lanes in which our sample is loaded. But in this moving boundary, because everything was mixed in the solution, so there used to be a lot of overlapping of the samples. Okay. So then they thought that why not we make a kind of a gel, and in gel we can have different lanes, and in different lanes we can add our samples individually to each lane. So what will happen? Now the samples will have a solid support to move also, and there would be no mixing of the samples, no overlapping of the samples, and therefore there would be increased resolution. So that they scientists thought that instead of having a moving boundary, let's have a solid support, right? So for that solid support, uh, yeah, they created another type of electrophoresis which is known as zone electrophoresis. Just a minute. So in zone electrophoresis, basically you have a supporting media, right? And therefore the purification and the analysis also is much easy with the help of zone electrophoresis. Now we can have this zone electrophoresis. May we, the best example is the gel electrophoresis we use. Now gel electrophoresis can be either horizontal gel electrophoresis or vertical gel electrophoresis. Horizontal gel electrophoresis is the agarose gel electrophoresis that we mostly use for nucleic acids, and for vertical gel electrophoresis, so horizontal gel electrophoresis, like this, the gel is there, the casting tray and everything, and vertical electrophoresis, which is your page, SDS page or page. So where there is an upper and a lower tank, and which is mostly used for proteins. So This is all what the cassette, the chamber, power supply, and everything looks like. And then these are some of the buffers. So first we'll solve one or two questions and then we'll come again to the division part. Okay. So the first question is, what is the difference between chromatography and electrophoresis? Electrophoresis can help to determine molecular weight, whereas chromatography cannot be used to determine molecular weight. Electrophoresis is used for the separation of nucleic acids, while chromatography is used for proteins. Sample run along the column with the help of a buffer in chromatography, while in electrophoresis, electric field is used to run the samples. So, what is the main difference between chromatography and electrophoresis?
Ma'am, option C.
Yes, option C is the correct answer. So, like we have just discussed, that the modification that was from chromatography to electrophoresis is initially that let the sample be in the buffer, but now we'll apply some electric charge also to it, and then we will have like a cathode and an anode. So we can separate the positively and negatively charged particles, and it would be easier for us. So like we have cation exchanger and ionic exchanger, it is more uh, like the resolution as compared to cationic and anionic, it was better for even for gel for moving boundary electrophoresis. Next question is, pick the incorrect statement regarding electrophoretic mobility. Electrophoretic mobility is directly proportional to the charge. It is inversely proportional to size and shape of the molecule, inversely proportional to the viscosity, and directly related to the mass of the molecule. Even if you don't know the formula, it's like very basic. You just have to apply some logic and you can get the answer.
Ma'am, option D.
Yeah. So now just like, okay, so here is the formula for electrophoretic mobility. Now this is the formula. You can have a lot of things over here, and you have some statements also, like here. These statements are in accordance with the formula. You can get the answer from here also, but the mobility of anything, if you have a particle of greater size and greater mass, obviously the its mobility would be lesser in comparison to a particle which is of lesser molecular weight and lesser size. So if I say that here you have to pick the incorrect statement, that electrophoretic mobility is inversely proportional to the size and shape, that is in general also any mobility is inversely proportional to the size. If the size is more, obviously the mobility would be less. If the viscosity of the medium would be more, what will happen? The frictional force would be more, so mobility would be less. If the charge is more, then obviously the mobility would be more. So, uh, in general, also you could have opted the answer, and obviously you have the formula also. Now, in moving boundary electrophoresis, sample is loaded on a solid support. Overlapping of the sample takes place, which give good resolution. Sample is dissolved in the buffer for the movement, or none of the above.
Ma'am, option C.
Sample is dissolved in the buffer for the movement. Yes. So C is the correct answer. So not many people are there today, maybe because we shifted the timing. But so we have just very few people. So now everybody has the opportunity to participate and everybody should try the answer the questions. Even if you don't know, it's fine. You can at least try. Okay. Now this is match the following. So try to answer this. Then we'll go to the PDF also to just have a quick revision. Temid, bisacrylamide, glycine, APS, tris HCl. So even if you are like not very sure, Even if you know like even one or two, you can figure it out. Can you try?
Yes, ma'am.
Tell Temid, what is the answer for Temid?
Ma'am, it's a catalyst, catalyzes acrylamide polymerization.
Catalyzes acrylamide polymerization. Okay, we'll just mark it one and then we'll in the end check. Bisacrylamide, cross
Cross-linking
linking, res. Glycine,
is present in running buffer,
okay. Ammonium persulfate, APS,
polymerization initiation.
yes. And tetracyclyl gel casting buffer. Now we'll just quickly see. So Temid, hmm. So Temid, it is used for the acrylamide polymerization. It catalyzes the polymerization. So you have to understand that ammonium persulfate is an initiator and Temid is catalyzing it. So there's a difference. It is starting and it is catalyzing. Then glycine is a component of running buffer. Bromophenol blue is a tracking dye. Do not get confused. And Comasi is a staining dye. So bromophenol blue is added for tracking and Comasi is used for staining of the gel. So tracking dye will always move faster than your protein or DNA. So whereas this is used for staining. So they have different functions. Then acrylamide, which is a monomeric unit which is used for gel preparation, and bisacrylamide is a cross linker for polymerization of acrylamide and bisacrylamide. So you have to understand. So basically, eh, depending on what kind of product, if we have any knowledge of the product that we want to resolve, we can change the concentration or we can change the pore size of the gel. So if you want gel of very high pore size, that means pore size should be very good enough, pore size should be very large, then you have to have lesser of your bisacrylamide and acrylamide because you want pores of a greater size. But if you want smaller pore size, then you have to increase the concentration of bisacrylamide so that your pore size are even smaller because there would be more bis, there would be more cross linking, there would be lesser size of the pores. Now we'll come to the next question. Yeah. So bromophenol blue and Comasi blue dyes are used for. So bromophenol blue is used for bromophenol.
Tracking.
It's a tracking dye. Here we have bromophenol is for tracking dye and Comasi blue is used for staining of the gel. So D is the correct answer. Then next question is, what is the function of glycerol during STS electrophoresis? It helps in denaturing the proteins. It helps in the loading of the sample in the well. It breaks the disulfide bonds. It helps in the movement of the proteins in the gel.
Ma'am, it helps in the loading of the sample in the.
It helps in the loading of the sample in the well. So, like if you don't have, so I don't know if you have done at least a gross gel electrophoresis. So always we have a loading dye. We have a loading dye in which we mix our sample. Like I'm talking about a gross gel electrophoresis because I do that often. So, in loading dye, whatever DNA is there, or if your PCR product is there, so we take some microliters of the PCR product and we take some microliters of the loading dye and then we mix it properly and then we add on to well. So if you do not add the loading dye which has glycerol in it, so what happens is that when you will load the sample in the well, it will come out. It will not stay there. It will just pop out from the well if you're not adding the loading dye, because there has to be something denser that can help it stably go there and stay there. So that's why glycerol, if it is not, your glycerol is not present, uh, sometimes what happens is that your loading dye, you're continuously using your loading dye and you're not properly mixing it. So what happens that all the glycerol content, it kinds of settle down at the bottom of your append off, and even when you are adding the dye, the sample will pop out. So therefore, always when you are doing any experiment, you have some solutions. For example, you have some dyes, or even if you have your DNA or protein or RNA samples, you should always mix it a little bit and then only use it. Because what happens is that the denser molecules tend to settle down at the bottom, and you will not get a properly mixed and a uniform content when you are pipetting it out if you're not mixing your samples properly. So what you can do, for suppose you have to put a PCR reaction, a general PCR reaction if you have to put. So you have your DNA and everything. So after thawing, so thawing always has to be done on ice. You cannot thaw your samples at room temperature on your workbench, especially when you're dealing with RNA, right? So after your thawing of the samples, you can give a short spin, like for 1 or 2 seconds, and then you can just very lightly tap it with your hands, with your fingers, your append off, and then you can use it. Or if you want to pipe it also, very gently you can pipe it up and down 2 or 3 times, and then you can use it. So pipetting is always important. So loading dye in a gross dye electrophoresis, if this is your tube, okay, and this is your loading dye. So loading dye means it has glycerol also, and it has a color dye also, just to make sure that your DNA has gone into the well, because DNA is colorless and glycerol is also colorless. So you might get confused whether your DNA actually get into the well or not. So that's why you have a loading dye. Loading dye, that means you have a dye along with your glycerol, maybe of blue or purple color, so that when you're mixing it with DNA and you're adding it into the well, you know that okay, my DNA got into the well now. So you should always mix your append off of the loading dye and then only take out the loading dye for your mixture. Otherwise, what happens? Glycerol will settle here at the bottom, and even you are taking the loading dye, your sample is popping out from the from the well, because it happened with me. That's why I'm telling you. Anyways, now, so running of the gel is something which is a little bit important when we are dealing with polyacrylate gel electrophoresis. So first we'll see here. So now what happens is, so when we're doing a vertical gel electrophoresis, that means we're uh, dealing with page. So we have two kinds of gels, okay? We have two different types of gels. First is your stacking gel, and second one is your resolving gel. Okay, so now we have studied over here about glycine, that glycine is present in running buffer also, and it might be present in the stacking gel also. It depends upon what kind of separation you are doing. Anyways, so we have two kinds of gels, stacking gel and resolving gel. So first we will load our resolving gel. We load the, if this is your cassette, you will load your resolving gel. Your resolving gel will come here. You will wait so that this resolving gel gets solidified, and then what you can do is, that depends what you are adding. So you can have a thin layer of any kind of organic solvent, or even you can have a thin layer of water also. It depends from lab to lab, and then you will add your stacking gel onto it, and then you will place the comb. This is the comb, this is comb, and then you will place your comb. So first, when you are preparing your gel electrophoresis, first you have to add the resolving gel. You will let your resolving gel to settle down. After the settling down of the resolving gel, you can add a thin layer of an organic solvent such as butanol, isopropanol, or some, in some labs they also add little bit of water over it, and then they remove the water just to make the surface smooth and just to make sure that everything is solidified. And then they add the stacking gel. Now, stacking gel, as the name suggests, stack. So, whatever sample you have added, so in the stacking gel, it will be properly stacked with each other. So that because it's a vertical system, so what may happen is one sample is here, another sample is here. So there can be a lot of distance between the two samples or two molecules or the two different kinds of uh, products. So just to stack them so that they can have like a thick layer, we use the stacking gel. And after your product, after your sample is stacked, then we run the resolving gel. So basically, what happens is that when you are running the sample, so once your sample is run on the stacking gel, you run on the resolving gel. So you just change the voltage. So when you see that the sample has run well through the stacking gel, you just change the voltage. So I'm a little confused about the voltage. So in one we use 60 volts, another we use 80 volts. I am confused in which one we use 60 and which one we use approximately 80. You can check it. So basically the difference between the stacking and the resolving gel while running is because of the voltage, because you change the voltage. But the basic chemistry behind what happens is that, see, first you prepare your samples. Your sample is prepared with the help of loading dye which has your SDS, which has your beta mercaptoethanol and glycerol. So beta, what is the difference between the function of SDS and the function of beta mercaptoethanol? Both are denaturing but in different ways. So beta mercaptoethanol basically breaks the disulfide bonds. Okay, so it will kind of break the polymeric chains into monomeric chains, into monomers. So beta mercaptoethanol breaks the disulfide bonds. And then glycerol, we know glycerol helps in the loading of the sample. Now, the difference here is that the pH of the stacking gel, here I have written, pH of the stacking gel is 6.8, whereas the pH of the resolving gel is 8.8. But how this difference in pH can make such a drastic change that your sample is stacking and then your sample is separating in resolving gel and it is getting stacked in your stacking gel? So what happens here is that when your sample is in the stacking gel, I'll just read it and then I'll explain you. Glycine is moving slowly in the front, whereas Tris HCl is moving fast. As a result, the sample gets sandwiched between glycine and Tris and gets stacked in the form of a thin band. So, I'll just write over here stacking gel. Okay. So, in stacking gel, what happens? Glycine, sample, and Tris. So, your sample get stacked in between glycine and tris. Here the pH is 6.8. Now we come to the resolving gel. Resolving gel. Now what happens in the resolving gel? As the sample enters into the resolving gel with pH 8.8, the glycine is now charged. Glycine is charged. It moves fast and now sample runs as per the molecular weight. Now what we are saying that in the resolving gel, the pH is 8.8. So glycine is getting charged. Now can anybody tell me how glycine is getting charged at pH 8.8?
Ma'am, the COH group is losing its H plus, so it will become negatively charged.
Yes. So first thing you have to understand is, so do you know about the concept of isoelectric points? So
Yes.
isoelectric point is basically a pH at which molecule has no net charge. So different amino acids have different isoelectric points. Okay. So if we talk about glycine, so the isoelectric point of glycine is somewhere around 6. Okay. The isoelectric point of glycine is somewhere around 6. Another concept is, I don't know whether you have studied or not, but you will surely study in your further classes. And this concept, this why I am explaining, it is not given in your this thing. It was not in the your lecture. But why I'm explaining is because this is the whole basis on the basis of which your glycine is used for the resolving. And this is something people, students never understand. Even I never understood why. What is, I just learned it, okay, in the resolving gel glycine will come forward because it will get negatively charged and then it will start resolving. But why it is happening, how it is happening, there is a logic behind everything in science, but we could never understand it. And also because this concept of pH and PI, it is very important for your like GATE and DBT and other competitive examinations. This concept is very important because they ask a lot of questions on this. So just a random, uh, if pH is greater than PI and if pH is less than PI. So if pH is greater than PI, then that particular uh, amino acid gets a negative charge. If the pH is less than PI, it is protonated and it gets a positive charge. So basically, when the pH is greater than PI, it gets deprotonated. There is a lot of explanation that is beyond the capacity of this class. But for as of now, you can remember these two uh, statements, that if the pH of an amino acid is greater than the PI, then it gets a negative charge, and if the pH is less than PI, it gets a positive charge. Now these two statements are very important. Why? Because you will have some questions in GATE where you have to calculate the net charge of the protein. So you will have a lot of amino acids, and then the question would be that you have to calculate the net charge of this amino acid of this protein. So there at pH, and they will write at pH this. So you need to have a basic idea about the PI values of each and every amino acid, and on the basis of it, you will calculate the charge on each and every amino acid, and then you will calculate the charge of the total protein. So this is a very important and a very like this question is asked often in examination. It's their favorite question, that calculate the net charge of protein at pH this, at pH that. So for that, you have to remember that what are the individual PI values of each and every amino acids, and then you have to calculate that at whatever pH it is given, whether that amino acid will have a positive charge or negative charge, and then you will calculate the net charge of that protein. So now, uh, at pH 8.8, now pH is above the PI of the glycine. So therefore glycine will be deprotonated and glycine will have a net negative charge. And since glycine is deprotonated, since glycine has a net negative charge, then therefore it will start moving fast, and therefore now your this glycine will come forward and your sample will start resolving. So you have to understand that why glycine start moving forward, why glycine is behind in the stacking gel, and why glycine comes forward and start moving in the resolving gel is because the PI of glycine is somewhere around 6. And when in the resolving gel, the pH is 8.8, and this pH is greater than the PI of the glycine. Therefore, glycine gets a negative charge, it gets deprotonated, which is obvious also if you go to all the basics and all. So therefore it starts moving. So this is how the difference of the pH creates this kind of variations, which helps in either stacking of your protein or resolving of the protein. So this was just like, I haven't even covered everything about, because it is, again, it's not, we cannot cover everything in just a two in just 2 hour class. So I just wanted to give like a brief about the electrophoresis, about the SDS electrophoresis. So SDS electrophoresis, even if it is in your coursework also or anywhere in your exams, so you really have to learn and understand and or just mug up the function of all the buffers and the reagents which are used, especially TEMED and APS are kind of important. And then you should know the basics of how the running of the electrophoresis happens in SDS electrophoresis. So these are two important things. Then we'll come to lecture 2, electrophoresis. So in this we have studied about, now that we have run our sample, the staining, different kinds of stains that can be used, the staining procedure, image analysis, and then there was a short video demonstration. So one thing that I would like to say here, that image analysis, so we will not discuss much about the demonstration and image analysis part, because that was kind of evident with the video, and also because there are many different softwares that can be used, and manually also you can do. So the procedure kind of depends on the software to software, but the basics remains the same. The basics is that you have to analyze the lanes. What are your lanes? If this is your gel, these are your bands. You have to analyze your lanes, different lanes. And so whenever you are doing SDS electrophoresis or this electrophoresis, so there is a lot of standardization that has to be done. Now, what is about this standardization is that how much sample you want to load, because what happens is that if you load a lot of sample, then your band would be very thick, something like this. These are called as smiling bands. So your band can be like this. Your band can come like this also. So your bands can come like this. Abhi, now if you want to calculate the size of the band, what will you do? Whether you will take from here or you will take from here. So the best way is either you take from the center, or you take from here also, you take from here also, and you take the average. But to avoid these situations, so this kind of smiling bands occurs. One of the reason is that you are adding a large amount of the protein. So it's always good to have clear, crisp bands like this. Although in Western it is not really possible every time to have good bands, but so that's why a lot of standardization is important. Another thing which is I'll just tell you on basis of my experience. So whenever you are doing research and you are doing Western blotting as a part of your research, or you're doing electrophoresis as a part of your research, so nowadays what happens is, ultimately when you do a research, your ultimate goal is to publish it. Not ultimate goal, but immediate goal is to publish it. Okay. So your immediate goal is to publish your work in a good journal, so that it can be accessible to other people, and also you have a kind of copyright that you have done this work. So nowadays what happens is that when you give your like whatever data you have, you give your manuscript for publications, so they ask for your original blots. What is the meaning of original blots? So like you did Western blot and you have a membrane on which you are uh, gel has come, so they ask for those original blots also, just to reconfirm that whether whatever data that you have mentioned in your manuscript is authentic or not. So they ask for the original blots. So why I am saying this, that now whenever you do any kind of Western, try to make a practice to store the Western blots of good results. So suppose if anybody of you, like some of the bachelor student, if you are doing some work in your bachelor's where some kind of Western blotting or some kind of electrophoresis is involved, and you think that you can publish your work in a good journal or at least in any general journal, then you should try to preserve the blots. Also, if you are in masters and you are aiming for PhD positions, this is a good practice when you get into PhD, to store your blots. It's a GLP, good lab practice, to store your blots, and not just your blot, to store any kind of raw data. So nowadays, like all the journals, they are very strict. They ask for raw data because now students are also very smart. They do a lot of manipulations. So not just learning science is important and learning good techniques is important. To be honest is also very important in science. You can have n number of ways to manipulate your data, but data manipulation will give you nothing. So if you get caught ki you have manipulated your data, your paper will be retracted. And believe me, retraction is the biggest black spot on your career. So if even your one paper is retracted, your entire career is spoiled. So that is why it's very important to be honest. Negative results are also results. So whatever results you are getting, whether it is in accordance with your hypothesis or opposite to your hypothesis, you should always be honest in recording it and telling it. Okay. So it is just like outside of the subject that we are studying, but just I'm telling you on the basis of my experience, if you really have an inclination towards research and you want to go into the research field. Anyways, so now we'll come to our questions, but just quickly we'll try to see what are the different staining procedures. So yes, so different types of staining. For the staining, different types of stains can be used. So we have Comasi, Comasi blue staining. It is a non-specific stain, okay? But mostly we use Comasi blue staining normally because it's easy to perform. So therefore we use Comasi brilliant blue. Then we have some other solutions like CBB. We have, it is a little more sensitive, 5 to 10 fold more sensitive is there. There is no de-staining step. So if you have gone through the videos, so it is I think in subsequent lectures that you have to do a de-staining step when you do your CBB dye this thing. So why de-staining is important? Because when you are putting your stain, everything is blue. Now what happens is that the interaction of the dye with the protein is greater than the interaction with another saccharides or something. So when you're de-staining it, only the proteins, the places where proteins are present, that will retain the stain, and all other places would be washed off. So de-staining will actually tell us that where the protein bands are present, because staining will make your complete paper blue, but your greater interaction of the stain with the protein, it will be there only at the places where proteins are present. So that is why in CBB you have to have a de-staining step. And suppose what happens at it one de-staining you are not able to remove all the stain, all the blue color, you can have a next round of de-staining step also. But in CBB, so CBB is more sensitive than your Comasi, and there is no de-staining step also. Then you have silver stain, and there are so many different stains available, but generally people in labs, they use Comasi brilliant blue only. Then just staining procedure, simple. You just remove the gel and place it in a container, and then you add your staining solution. And then so whenever you are doing SDS and you are doing Western, so there is a shaker. On shaker you have to put your gel, And then when you add your dye and you put your gel onto the shaker, so slowly and slowly it will stain your gel. So that is why it's not like that you just took out the you have taken out your gel, you poured it into the staining solution and immediately you're taking it out. No, you have to put it into the shaker, for about 30 to 60 minutes, for 40 minutes on an average, so that everything is properly stained. And then you discard the solution and then you wash it and then you add your destaining solution and then you again kind of mix it on a shaker. And then if the color of the destaining solution is intense blue, replace it with the new destaining solution. So up till now what we have done, so now we know that after chromatography we there is another way by which we can determine the molecular weight of protein, and that is electrophoresis. So electrophoresis, it can be a moving bound electrophoresis or a zone electrophoresis. Moving bound electrophoresis means that there is no solid support requirement, but in zone electrophoresis solid support is needed. Then you have vertical electrophoresis and horizontal. Here we are talking about vertical electrophoresis. In vertical electrophoresis, you have a stacking gel and a resolving gel. Stacking gel pH 6.8, resolving gel pH 8.8. This difference in pH will cause the difference in the mobility of glycine, and therefore your sample is able to stack or resolve. Then now that you have completed your electrophoresis, your sample has run. Now you want to stain it to see what are the places where protein bands has come. So for that you have to stain it. You stain it with Comasi blue and then you have to destain it with the help of destaining solution. So electrophoresis we are talking about. Hmm. So we talk about basic of electrophoresis. I'm really sorry for my bad handwriting. So you have to prepare the sample. So sample you have to prepare in loading dye, and you have to add loading dye which has SDS, beta mercaptoethanol and glycerol. After preparation of the sample, you have made your resolving gel and stacking gel. You first add your resolving gels, and then you add your stacking gel, and then you add your comp. And when your gel solidifies, then you add the sample, and first it will get stacked and then it will resolve. After the resolving of the after the run is complete, you will take out your gel and then you will put it for staining. Now we'll see some of the questions. So the composition of the destaining solution is methanol and glycine, glycine and acetic acid, methanol and acetic acid, or none of the above. What is the composition of destaining solution?
Methanol and acetic acid.
Methanol and acetic acid. Right. Then manual lane detection is recommended when bands are clear and straight, bands are non-overlapping, bands are smiling and overlapping, and bands are very light. Manual lane detection.
Option C.
Bands are smiling and overlapping. Yes. So we'll just quickly see about the image analysis part. So the thing is that when you are analyzing your bands, so if this is your uh, gel or gel where you have all the run or the membrane, now these are your different lanes, and these are your bands. So when you're keeping it in a software, so the software can detect the lanes. Okay, software can detect the lanes. But sometimes what happens that your bands can be overlapping. Like this is one band and somewhere here it is another band. So software might not detect this and it might detect it as just one lane. Or if the bands are smiling like this, they are very thick like this, and another band is like this or like this. So when the bands are very overlapping or very smiling bands, then the software cannot detect the lanes. So you it is advisable to detect the lanes manually when your bands are not very clear and distinct to each other. So we'll just quickly see. Yeah. So you can just see here that when you stain with Kumasi, so what happens? Everything is blue. But when you destain it, then you kind of have your bands. So this is a very good electrophoresis where you can see that there are no smiling bands also. So that means that it is a well standardized protocol that they have used. They have already standardized how much protein has to be added, what should be the voltage for how much time it has to be run. So like this, your Western blot should be there. Then, so just quickly we'll see the image analysis part. So in the image analysis part, so always you have a marker. So marker means ladder. So either you are doing a gross or you are doing SDS page, you always have to have a marker or which is known as ladder also. So what does a ladder or a marker means? So suppose uh, you in your marker, so basically you have bands at definite locations that are already known. So what is the meaning of this? That suppose this you have run your ladder sample in one lane and your sample in another lane. So you get a lot of bands 1, 2, 3, 4, 5, 6. But this marker is provided by a company. So companies, it is a professionally made marker. So it is already known that this band will correspond to this band will correspond to this weight, this band will correspond to this weight, this band will correspond to this. So this information we already have. So it's just like when you do if you have done some spectrophotometric technique or in any kind of technique, first you have some standards. So first you run your standards and you make a curve or you make a line and then you run your unknown sample. So on the basis of where your unknown sample has come, you compare it with the help of the standards that you have already run, and then you kind of calculate the molecular weight. So basically marker gives us a comparison key if my band has come here and it has come between this and this marker and this band corresponds to this and this weight. So I can have a knowledge that okay, my band will have approximately this much of the weight or my protein will have approximately this much of the weight. So that means you have to have a control sample on the basis of which you compare your unknown. So you have to have some known samples, some known well-defined standards which are used for comparing and which are used for identifying the unknown sample's molecular weight. And this concept of running a known sample is used everywhere in biotechnology. It is used in agros also. In agros also you have a ladder. You have different ladders. So when you run an agros electrophoresis, so there also you have ladders like ladder of 1 kb ladder, 50 base pair ladders, 100 base pair ladders. So what is the meaning of this? That it will resolve up to 1 kb, it will resolve up to 50 base pair, it will resolve up to 100 base pairs. So you have different bands like this. This band will, suppose it signifies 500 base pairs, this signifies 1 kb, this signifies 1.5 kb, 2 kb, 3 kb, 5 kb. So like this. So you run your ladder here and then you run your sample. And suppose your sample gave a band somewhere around this, and you compare with the help of ladder, you saw ki acha, this band is a 2 kb band and my sample is somewhere around this. That means my sample is also of 2 kb size. So this way your ladder is used to identify or know the molecular weight of your sample. Even in the case of agrostory electrophoresis, and same is done with the in your SDS electrophoresis also. Even when you go for sequencing, so I don't know how many of you have any knowledge about next generation sequencing, but even in next generation sequencing, you have to have a library or a known genomic library which is used as a standard on the basis of which you compare. So this running of standards, running of known samples is used everywhere. Anyways, so after this, for your image analysis, so there are so many softwares which are used: VisionWorks, Total, Lab Series, GelPro, NIH, and all these. So different softwares are there for the analysis of the image. So for the first step in image analysis is you have to detect your lane, which lane is there. So you have to detect all the lanes, you have to mark the lanes, and this lane can be detected automatically by the softwares, or you can do it manually also. Then you have to select the bands. So select the bands means, so if you have if you have gone through the video, so there you have you must have seen key there are so many bands are there. Many bands come even in one lane. So I'll just, it's like, hmm. So even in one lane there are so many bands. So many bands even in one lane. Some are very light bands and some are very dark bands. Some are dark bands and some are very light bands. So now, according to your interest, you have to select which band you want, whether you want a darker band or you want light bands. So you have to detect your bands also, you have to select your bands also, you have to then remove the background, like whatever bands you don't want you can delete it. And so this all is you can do with the help of your softwares, and then you do the measurements. So this is your week lecture 2 of the week 9. Now we'll come to the different variants of page. So this vertical electrophoresis, yes. So this vertical electrophoresis can have several variants. Okay, we have already discussed about SDS page. So basically SDS, sodium dodecyl sulfate, is an anionic detergent which imparts negative charge to the proteins, to the individual amino acids. So this SDS will cause the denaturation of the proteins. That means the native conformation, the native order in which the DNA or the protein is present, it is destroyed. So in SDS, you cannot detect the 3D, that means the three-dimensional structure of the protein, because your protein is denatured. But there are some other variants where The protein is not denatured, and therefore you can have an idea about the 3D conformation of your protein, and that is your native page. So basically what happens in native page, so in SDS page you have your anionic detergent, that is sodium nodecyl sulfate and beta mercaptoethanol. So this beta mercaptoethanol, it breaks the disulfide linkages, and your sodium nodecyl sulfate, it imparts negative charge to the proteins. So the 3D structure of the protein is destroyed, and therefore the movement is only on the basis of molecular weight, because each and every protein, each and everything has negative charge because of SDS. So basically, your mobility depends upon charge by mass. But here, in the case of SDS page, because negative charge is imparted because of SDS, the separation is only on the basis of molecular weight and not on the basis of charge. Whereas in the case of native page, the separation occurs on the basis of charge to mass ratio. It's not only charge, it's not only mass, it is the charge to mass ratio. So therefore, if you suppose if you have just a minute, anyways, yeah. So suppose if you have uh, one protein which is having a positive charge overall, and one protein which has a negative charge overall, so you can ask whether they will move in opposite direction or the same direction, like how they will move. But it's not only the charge that matters, it's the charge to mass ratio that will determine its mobility. Then this is your native page. Then you also have urea page. So basically, urea page is also it will also kind of denature the protein. But so what is the difference between an SDS page and urea page? Can anybody tell me how SDS page is different from urea page? Because in SDS page also denaturation is happening, and in urea page also denaturation is happening. Then why to have two different variants of page where both are providing us denaturation only? Why to have extra urea page?
In STS page, the negative charge is there, but in urea page, there is no negative charge imparted to the.
Okay, that is one of that can be one reason also, and another reason also is that so basically, uh, just a minute. Yeah. So you have to understand that when you have DNA, okay, from DNA you have your we're talking about proteins, we're talking about mRNA. This is known as transcription. Okay, from mRNA what is made? Proteins. This is your translation here. Now this protein, whatever protein that is made, it's not the end of it. Why? Because I think we have already discussed it also, that this protein has to undergo several modifications. Okay? Several modifications. So this protein, not you just so you just not have to synthesize proteins, you also have to fold it into proper conformation. Then only a protein is able to do its function. Then only you have a proper protein. So it's not only the synthesis that is important, but the proper folding of proteins. It's very important. Proper folding of proteins. So proper folding of proteins is equally important as the synthesis of proteins. So that is why you have we have already discussed in previous classes also that after the synthesis of the protein in the endoplasmic reticulum, protein undergoes various types of modifications like N-linked glycosylation occurs, and then in Golgi operators also O-linked glycosylation occurs. So these glycosylations and other modifications kind of help the protein to be in a properly folded form. Also we have chaperones. I don't know whether you know about chaperones or not. So, do you know about chaperones?
Yes, ma'am.
So, what are chaperones?
Ma'am, they have to hold the proteins for scaffolding.
Yeah, so different kinds of chaperones have different functions. So, in general, so basically, chaperones, what happens is that they prevent the misfolding of the proteins also, and they help in the proper folding of the proteins also. So, there are different kinds of chaperones. Some chaperones prevents the misfolding, and some chaperones helps in the proper folding. So, why I'm telling you is that protein synthesis is a very intricate process. It not only involves the synthesis of the proteins and individual amino acids, it also involves the proper folding of the protein. So, suppose if you're doing any experiment which involves protein isolation and purification, then you have to make sure that your only objective is not just to isolate the protein, but you have to isolate the protein in its correct conformation, in its correct folding also. That is why when you are doing any kind of protein downstream processing, which involves proteins, obviously, you have to make sure that it is always better to have lesser number of purification steps, because more the number of steps, more is the chance of degradation of the proteins. Now, one very important reagent which causes the unfolding of the protein is urea. So urea is responsible for the unfolding of proteins. So, if you want to study about the folding status of a protein, or you want to have the information of how the protein has folded, then you can use urea. I think we'll be studying also in later on in this class only, that how urea helps in understanding the folding status of proteins. Okay, so basically urea, the different concentration of urea will help to have a knowledge about the sequential folding status of proteins. You add some concentration of urea, protein gets folded. You increase the concentration of urea, protein is folded even more. Then you are also increasing, then again protein is unfolding. So like this, your unfolding of the protein, sequential unfolding of the protein is helped with the is can be studied with the help of urea. So basically, here also separation is occurring on the basis of charge to mass. Why? Because SDS was imparting negative charge, but urea is not imparting any negative charge. So separation is also on the basis of charge to mass, but you can have the knowledge of the folding status of protein. Okay, so the difference between urea page and SDS page is that one difference is okay, that SDS is giving negative charge. So here separation is occurring only on the basis of molecular weight. Here separation is occurring on the basis of charge to mass ratio. Okay, that is fine. But the major difference between SDS page and urea page is that you cannot study the folding status of a protein with the help of SDS page. But you can study the folding status of the protein with the help of urea page. Okay, then there are many applications, so we'll just try to see one or two questions first, and then we'll come to the description part. So in lecture 3 we have studied about the different variants of page that we have already discussed just now. So different variants are your SDS page, your native page, and your urea. So urea page helps to study the folding status. Okay, here you can study the 3D conformation, and this is a normal. Here separation occurs on the basis of molecular weight. Here charge to mass ratio. Here also charge to mass ratio. Only in SDS charge does not contribute because SDS imparts negative charge. So this is about the different variants of page. Then we'll see what are the applications of page, and then Western blotting. So Western blotting is also very important when it comes to your experiment point of view. Western blotting is a very important technique. Also, questions also come But more questions in your like if you're focusing on competitive exams, more questions would be on electrophoresis. Anyways, the difference between native page and SDS page is native page preserves the 3D structure while SDS page does not. Native page runs only by charge and SDS by mass. Native page separates through charge by mass via SDS through charge, all of the above. So what is the answer?
Mom, option A.
Native page preserves the 3D structure of the protein. Yes. So option A is the correct answer. Here, difference between SDS and urea pages. SDS denatures the protein while urea does not. SDS impart negative charge to protein while urea does not. SDS separates the protein on the basis of mass and urea by charge. And urea imparts that positive charge. So separation is through mass. So on the basis of these options, what is the correct answer?
Option D.
SDS imparts net negative charge. Yes. So both are denaturing proteins. This cannot be the right answer. SDS separates a protein on the basis of mass. Yes. And urea by charge. No. Urea separates by charge to mass ratio. And urea imparts net positive charge. No. Nothing like this. Then next question. Okay. So before these questions, or we can just try these questions first. Okay. So uneven heating of the gel results in diffusion of the protein bands, distorted protein bands, smiling bands, or double bands. Uneven heating of the gel. What is the answer?
Option B.
Distorted protein bands. Let's see. You said option B, na?
Yes, ma'am.
Distorted protein bands. But uneven heating results in smiling bands. So here you can see that these are actually the troubleshooting. So the answer is smiling bands and not distorted protein bands. So uneven heating of the gel causes differential migration of the proteins. So what happens is uneven heating of the gel. This is your gel, okay? Let us think that I have zoomed one lane. This is one lane, and I have zoomed it here, and this is one lane I'm talking about, okay? And in this lane, in this part, and in this part, it is very heated up. So here movement is more. Here movement is more. But this middle portion, the gel is not that much heated up. So here movement would be less. So you will get a band like this. Could you understand? See, if you have a band like this, that means this portion and this portion has moved faster than this portion, because this portion was heated more, therefore it moved faster, and this portion was heated less, therefore it moved slower. Similarly, if you have this band, so that means the middle portion, the middle lane was heated up a lot, and these portion was not that much heated, therefore you get this type of band. So there was uneven heating of the gel. So uneven heating of the gel results in smiling bands. Could you understand how smiling bands are formed because of uneven heating?
Yes, ma'am.
Because uh, in the same gel, these 2 points are getting heated less, and this point is getting heated more. So if this was your sample which was loaded, so this portion moved more, and these portions moved less. Similarly, in this case, here you can see that heating was here more. So if this was your initial sample, so these portion moved more, because it was heated a lot, and this portion could not move that much because it was not that properly heated. Therefore, uneven heating results in smiling bands. Next question is, lateral spreading of the bands can be avoided by increasing the amount of APS, preparing fresh samples, running the gel at lower current, or reducing the time between loading of the sample and running. Think, think what can be the answer. Okay, we'll just see over here. Okay, so how smiling bands are formed, this is clear. Okay, now we'll see one by one what different kinds of bands that are formed, what are the reasons. So diffuse bands, how diffuse bands are formed, matlab how that they have not written. Okay, diffused bands can be corrected by increasing the concentration of acrylamide. Yeah, obviously, because so basically acrylamide will increase the pore size, will reduce the pore size, and therefore you can have lot of sharpness and crispiness in the bands. So you will not have diffused bands. Okay. Here the question is lateral spreading. So we'll see how lateral spreading can be avoided. Yes. So if the protein bands appear laterally spreading, it can be avoided by reducing the time between loading of the sample and running. So how many of you have loaded sample either in agarose gel or in normal SDS in any of the gel? Have you ever loaded sample anywhere in agarose gel also or in SDS? Has anybody ever loaded samples? Nobody has loaded. Nobody has done agarose gel or this thing SDS.
Not yet, ma'am.
Okay. Okay, fine, no problem. So, what happens is that suppose I have prepared my gel. Okay, I'll explain with agarose gel first, and then I will come to late SDS. So, this is your agarose gel. It's a horizontal gel and SDS gel. Okay, so this is an agarose gel, and these are my different lanes, and these are my wells. I load the sample here. I loaded the sample, and then I went to canteen, and then I had my snacks, and then I came after after half an hour, and then I thought, okay, chalo, I will run the gel. What will happen? The DNA or the DNA with the loading dye that I have added, it will get diffused all over. So if this was the well, and here I have added my DNA along with the loading dye. So after half an hour, I will see it like this. It will diffuse. Why? Because this gel has pores. So when you are providing electricity, so they are moving. When there is no electricity, obviously they won't move that much, but there would be diffusion. So because of the diffusion, my band would look like this. Therefore, whenever you are doing any kind of electrophoresis, and similarly here also, you are adding, and because it's a vertical, so you will have a lot of diffusion like this. So therefore, whenever you are doing any kind of electrophoresis, immediately after adding the samples, immediately you should turn it on for running. Or if someone says that, okay, I have 5 samples and you have 1 sample, so you load your sample and then I will load my sample and then we can run the gel. So what you will say, that first you load your 5 samples and then I will load 1 sample. Why? Because for loading 5 sample it will take more time. So if you have loaded your sample first and then he will load your 5 samples in the other lanes, then your in that time your sample will start getting diffused. So therefore, whenever you are doing agross electrophoresis, you should be very quick. Quickly you should add the samples and quickly you should start your run. You should not wait, or you should not be very lethargic and slow. That you are adding slowly you are adding your, slowly you are putting one DNA in one well and then you are talking and then you adding in another well. You should not do like this. You should be very quick enough to immediately add the sample in all the wells and then immediately you put it for this thing, run. And always there is a tracking die. Okay, there is a tracking die. So tracking die in the case of SDS, if this is your sample, tracking die would be somewhere here. So tracking die basically is used to track the run, that how much your sample has run. So basically, um, if you have an agarose gel and this is the, suppose this is the length of the gel tray, your tracking die should travel more than 50%, and here also your tracking die should travel more than 60%. At least it should come here, then only you have to switch off. Because if your tracking die is here, then your DNA would be here somewhere. Or if your tracking die is here, then your protein would be somewhere here. So therefore, your tracking die should always travel more than 50% in the case of agarose gel. Here also at least it should travel, in SJs also it should travel approximately 60 to 70%. Tracking die should travel. And suppose if you have difference of very less base pairs, suppose you want to resolve 2 bands, one band is of 150 base pairs and one band is of 200 base pairs. Okay, so the difference is only 50 base pairs. So it's a very small difference as compared to, let's say, 2 bands, which is in which one band is of 1 kb, another band is of 8 kb, say. So it is a lot of difference, but here difference is very less, and we have even lesser difference bands. So in that case, you should always run the gel for a longer time, at least for 70%, so that these bands can properly resolve. Otherwise, these bands will not be able to properly resolve. Is it clear to everybody?
Yes, ma'am.
So diffusion happens when you are keeping your sample along with your loading die in the gel for a very long time, then diffusion happens. And there are other also this sorry, yeah, you can have distorted protein bands also. So distorted protein bands uh, are because of the stacking gel polymerization, which can be corrected by increasing the amount of ammonium persulfate or TEM. So like these all things are there. You just need to have like a basic read and you will get through it. The only 2 points which were important is lateral spreading and smiling bands that we have already discussed. That's why I gave questions about these 2 bands. Also diffused protein bands are also occurring, so that you can also, so there are many things. So when you are running an SDS page, so it's not just following the protocol. So in experiment, when you are learning something in your notebook, the principle and your experiment are two different things. So in principle you will have a protocol that you have to do this, you have to do that, and then you think that okay, after following all the steps my result will come. But it never happens in reality. You have to standardize it. So every protocol needs standardization depending upon the kind of reagents you are using, the kind of the hand, because you have the same, even in the kitchen, like we have the same spices, but the food that we cook is tastes different from different hands, because handling is different. Also, the addition of the spices is different. Some adds more, some adds less spices. So same happens in the case of research also, that you have the same protocol, but if two different people are performing, then they will not get the same results because handling is different. So therefore, whenever you are doing any experiment, here we're talking about SDS or any experiment in general, you have to standardize it, and not just standardize it, you have to standardize it in a manner that it is reproducible. Reproducible as in that if you are getting results with your handling 80%, then at least for 50% it should be present when someone else is doing the same experiment following your protocol. So it is very important even when you are publishing a paper. Anyways, now we'll come to the applications of page. First, we'll solve one or two questions and then we'll go to the applications. Okay, we have already studied it, very easy. The pH of stacking and resolving gel in SDS page is, stacking gel has a pH of
6.8.
6.8 and resolving gel has a pH of,
6.6.
no, resolving gel has a pH of
8.8.
6.6. Why you think 6.6? We have 6.6 anyways is not even in the option, but we have discussed now that the pH of resolving gel is more, therefore glycine which has a pi, glycine which has a pi of approximately 6, and because the pH is more than pi, therefore glycine has a negative charge, therefore glycine moves faster, and therefore your sample gets resolved in the resolving gel. So always remember, the pH of resolving gel is more than that of the stacking gel. Okay, if you have, suppose you don't remember कि 6.8 किसका है 8.8 किसका है, but you, if you know only the basic key, the pH of resolving gel is more than that of stacking gel, then you can just confirm that okay, if this is 8.8 and 6.8, but I know that resolving gel has a greater pH, so 8.8 would be of resolving gel, 6.8 would be of stacking gel. Anyways, we come to the next question. Function of beta mercaptoethanol in SDS page is to give negative charge to amino acid in proteins, for the oxidation of disulfide bonds, for the reduction of disulfide bonds, and for breaking hydrogen bonds. What happens?
Option C.
Yes. So we have already discussed that beta mercaptoethanol, it is used for the reduction of the disulfide bonds. Okay. Now, next questions. In SDS page of protein separation, one SDS molecule will bind to every amino acid, every 2 amino acids, every 3 amino acids, every 4 amino acids. So one SDS molecule binds to how many amino acids in SDS page?
2 amino acids.
2 amino acids. So one SDS will bind to, I'm sorry for the noise which is coming, but I do not have any control over it. Okay. Next question is, with identical molecular weight, which DNA will migrate faster during agarose electrophoresis? Supercoiled, single, double-stranded, nicked. Any idea? I don't think we have covered it. Just
From A.
supercoiled. Okay, can you explain how and why?
I think it's in the most compact
Will
profile.
single stranded DNA travel faster than double strand?
Uh, what are you saying? Dual-stranded DNA. Why you say so?
No, because double stranded will take a bit time, right?
Sorry. There's a lot of noise coming here. Yes.
Uh, like double stranded will be a bit heavier than the single one, right?
So, a single stranded DNA will move faster than the supercoil, but that single stranded DNA has to be circular. If you have a
Yeah.
circular single stranded DNA, then it will move faster than the supercoiled DNA. But if you have a linear DNA, then supercoiled DNA will move faster. Okay, because here it is all on the how much compact your DNA is. So suppose if you have a supercoiled circular DNA which is double stranded and a circular single stranded DNA, then your circular single stranded DNA will move faster because both are compact, but because it is single stranded, therefore it will move faster. Here I have already only written single stranded, so here it is. In your if these kinds of questions are there, then you have to choose the best possible option, because I haven't mentioned whether it is double stranded, sorry, circular single stranded DNA. I have not written whether it is circular or linear. So therefore, here according to the options, it should be supercoiled DNA. Okay, so it's like so if you have,
Ma'am,
yes,
and what about the nicked DNA?
yeah,
Like,
I'm
what is
telling.
the difference between that?
So this is your circular but single stranded. Okay, you will have your supercoiled, you will have your linear, you will have your nicked. So, or some relaxed DNA. Relaxed, so this is how your, I mean, it is like this. So this is the first one and this is the least. So you can note it down if you want. Like this is how the separation occurs, because it all depends on how compact your molecule is. For okay, I'll give you some example. So if you have threads, okay, so if you have threads which are intertwined with each other like a ball, which is kind of a supercoiled DNA, and if you have long threads which are not intertwined, they're just straight long threads, then, and so what is easier for you to move? Your supercoiled or intertwined thread is easier to move in comparison to the linear long threads if you have woolen threads, let's say. So same kind of principle applies to DNA also. Like to roll a ball is much easier than to roll some kind of linear structures, because the ball is, because it is supercoiled, because the it is so compact, so it is also experiencing, there is less surface area for the frictional force also to apply, and therefore the movement would be more, and therefore your supercoiled DNA will always move faster than your linear DNA. And when you are having a linear DNA which is even nicked, so you know what is the nicked DNA? Nicked means that this is a DNA, it is nicked from here, that means it is like this, somewhere it is nicked, or you have a relaxed circular DNA. So what is a relaxed circular DNA? Anybody has any knowledge on what is a relaxed circular DNA? Okay, I'll tell. So relaxed circular DNA means, so um, um, have you studied about replication?
Yes,
Yes,
ma'am.
ma'am.
So have you studied about some enzymes which are called as topoisomerase?
Yes.
So what is the function of topoisomerase? It removes supercoiling. Okay, so before DNA replication, your DNA is so much supercoiled, so it removes your supercoiling. So basically, your relaxed circular DNA is a DNA where your supercoiling is removed or supercoiling is less. Because of the nick, there is supercoiling is also less. So basically, there is one number. I don't know whether you have. It's actually going a lot beyond your linkage number. Do you know? Have you any idea about linkage number?
Yes, ma'am.
What is the linkage number? It is
Ma'am,
the
it's total number of rights plus
huh,
वैसे कुछ तो
plus
twist.
twists. Okay, so basically, and you must have also studied that this can be linkage number, how it can be increased, how it can be reduced. So you have to have a nick. So link basically kinds of reduces the supercoiling, and when the supercoiling is removed, what will happen? The DNA will become less compact because supercoiling is causing it to become more compact. Supercoiling will increase the compactness of DNA, whereas decrease in the supercoiling will decrease the compactness of DNA. Therefore, a relaxed circular DNA, which has less supercoiling, will be less compact. And because it is less compact, therefore it will travel very slowly. Okay? So Nick kinds of removes the supercoiling, makes it relaxed, and therefore it reduces the super compactness, and therefore it travels least. So anyways, what we were doing, yeah. So yeah, so the answer is supercoiled circular DNA. Now we'll just quickly go through the applications of page. We don't have much time also. Uh, okay. So determination of oligomeric status of protein. So basically, what is the meaning of oligomeric status? I think we have already discussed one question related to it, how to determine the oligomeric status, whether the protein is in a monomer or heterodimer or homodimer form. So oligomeric status can be determined. It's a simple formula: molecular weight in native and molecular weight in SDS. So because in SDS you will have different subunits. Okay, so because you are also using beta mercaptoethanol, so beta mercaptoethanol will remove the disulfide linkages, and SDS will obviously denature the proteins. So and in SDS we get a whole molecular weight, and in native we get the whole molecular weight, and in SDS we get the molecular weight of the individual chains, let's say. And therefore by uh, dividing that, you can get to know how many units are present. 5 are present, 4 are present, like that. Then we have discussed that page can be used for determination of the folding status. So with the help of urea, how we can use to determine the folding status. So as we are increasing the concentration of urea, the protein starts getting unfolding, and when the protein starts getting unfolding, then the migration would also differ. And when the migration will differ, you can see here native protein and unfolded protein. So again the logic is same, that when something is getting unfolded, it is not that much compact. The compactness is getting decreased when the protein is getting unfolded, and because the compactness is getting reduced when the protein is getting unfolded, therefore the migration is also less. Native proteins are able to migrate more because they are very compact, they are compactly folded. But because you are increasing the concentration of urea, your protein is getting unfolded, and therefore you will see key now the protein here you can see now these three bands are same. That means now the protein is maximally unfolded, it cannot unfold more than this. So like this, you can, with the help of migration, you can determine whether the protein you can determine the folding status of the protein, because unfolded protein will migrate less in comparison to your native protein. Then Then you can have protein protein interactions, other kinds of interactions. Western blotting, now we'll see Western blotting. So, can anybody tell me that why Western blotting is needed? From electrophoresis, we have now the uh, run. So, how blotting will benefit the electrophoresis? Anybody has any idea?
It helps us to identify the protein by antibody antigen reaction. So if with the electrophoresis, we only get to know what the molecular weight is, and if we use Western blotting, we get to know the specific protein, like which protein it is.
So, can't I do the antigen antibody interaction in the gel? Like, why I need to put it in a put it on a membrane? Can't I add directly the whatever whatever interaction I want to find out? Can I do it in the gel itself?
No, ma'am.
Why?
Gel is not that much stable as in compares to nitrocellulose membrane or.
Yes. So that is the most important point. That when you have run your sample, okay, you have run your sample. Now you know that these are the proteins. Now the gel that you have is very flimsy. Okay. So it's not very easy for, also, like as I just mentioned, that nowadays you need raw data for your publications and all, and also for your further work, whatever further experiments that you have to do, you have to perform, you cannot do on the gel because it's very flimsy, it can easily break. So it's always better to, for all the further analysis, for all the further experiments, that if you kind of can have a replica of the protein of the bands on a paper, on a sheet, either it can be a nitrocellular membrane or it can be a PVD membrane, PVDF membrane. So, so that is why. So, Western blotting helps. Blotting means you're blotting, to blot. So, we have different kinds of blotting also. So, what are the different kinds of blotting? Northern blotting. So, why another? How? What is the use of northern blotting?
No, for DNA.
And for RNA, then what we use?
Okay. Southern for DNA, northern for
Huh.
RNA, and western for
So,
protein.
yes. So, we have southern blotting. So, southern blotting is used for DNA, and northern blotting is used for RNA. We don't have an eastern blotting as of now. So, anyways, so basically, what you are doing in western blotting is that whatever protein that you had in your gel, you are transferring it to a solid support such as a nitrocellular membrane or a PVDF membrane. So that is what they have written, that transfer of protein onto a solid support such as a nitrocellulose membrane, and then you can have your antigen antibody interactions. Okay, then you can put your whatever protein of interest you have, then you can add your primary antibody and secondary antibody. So what is the difference between a primary antibody and a secondary antibody? Can anybody tell?
The primary antibody binds to the antigen straight away, and the secondary antibody binds the primary antibody.
Yes. So, primary antibody is specific to the protein, but secondary antibody is specific to the primary antibody. So, why you think that we need a secondary antibody? Sorry for the noise. So, why you think we need a secondary antibody? Why can't we just have a primary antibody and detect the signal? Why we need a secondary antibody at the first place?
But the use of secondary antibody makes sure that we don't have to use much of the primary antibody, and also the result is better when we use secondary antibody, as the label and all, we are not directly putting it on the primary, so it will not affect the interaction between the protein and primary antibody.
Okay, good. So this is very important reason. There's another reason also. So I'll just tell you. So what he has said is absolutely correct. So one thing is that primary antibodies are costlier than secondary antibodies. Secondary antibodies are cheaper than a primary antibody. One thing. Second thing is that after your primary antibody, whatever fluorochrome, or if your primary antibody is also attached to a fluorochrome molecule, that it can hamper the interaction between the antigen and the antibody. And also one very important point, which is actually the most important point, is that we use secondary antibody for the amplification of the signal. Okay, amplification of the signal, to amplify the signal. So basically what happens is that so when you do microscopy, so already when you do microscopy, when you are acquiring your data on a fluorescent microscope or in a confocal microscope, so in their software looks very beautiful, very nice, and as soon as you take your images from the microscope to your laptop, the contrast immediately reduces a lot, and you will not have very beautiful pictures, and you might not be able to detect some minor intricacies or like differences in the localization of certain antigens and proteins. So therefore, to have a better understanding, to have a better image, when you are using secondary antibody, it will amplify the signal. And when the signal is amplified, the contrast will also increase, and you will have more, you will have better images. And also, like he said, that if this is your antigen, okay, and this is your antibody, it is binding to it, and you have a fluorochrome molecule which is attached to the antibody, then this fluorochrome molecule, it can hamper this interaction. But if you have another antibody which is binding to this antigen, and then you have a fluorochrome molecule, then there is no chance that this interaction is getting hampered. So therefore, you have a secondary antibody. So this concept of having a secondary antibody and primary antibody applies to every antigen antibody interaction. It applies to fluorescence also. It applies to ELISA also. So wherever you study about antigen antibody interaction, the concept and the logic of using primary and secondary antibody remains the same here also. So basically, protein is an antigen. I mean, antigen is made up of proteins. So basically, antigen antibody interaction means antigen means protein. So protein antibody interaction is what we actually study over here. So if you haven't studied, you should go through the antigen antibody interactions. It's a basic concept which is used in many places. Okay, quickly we'll do these 4 or 5 questions. So disulfide bonds are formed between cysteine residues, threonine, proline, histidine residues. We have already studied
Cysteine
cysteine
residues.
residues. Then cleaving of peptide chain is done by
Trypsin.
trypsin. The function of glycerol in Page experiment is stabilize protein structure, helps to bind SDS, helps to reduce disulfide bond, provide density to the sample.
Option D.
Option D. Someone else was also saying something?
Yeah, option D. Density.
Yeah, right. So when you have your sample becomes denser, so it is able to go into the well and it is not popping out of the well. Otherwise, if you don't add loading dye which has glycerol in it, your sample will not remain in the well. Okay. Last question. Charging of PVDF membrane is done by glycine treatment, immersing the membrane in 100% methanol for 30 minutes, immersing the membrane in 100% methanol for 30 minutes, and then in transfer buffer, and none of the above.
No, option C.
Yes. So PVDF membrane charging happens by immersing the membrane in 100% methanol for 30 minutes and then in transfer buffer. So here also, I think just quickly we'll see. Yeah, so Western blotting, first you have to prepare your sample. So, I mean, this preparation of sample basically is for the electrophoresis, that you have to first prepare your sample, and then you have to run your sample, and then you have to transfer it into the membrane. And charging means that basically your nitrocellular membrane needs not to be charged because it is already hydrophilic, but PVDF membrane has to be charged. And then you have to assemble the cassette. You have to put, like, you have some many kinds of filter papers and buffers and blotting sheets. So there is a sequence of how you have to put first the filter paper, and how you have to put the gel, and then you have to put membrane and all that. And then after the assembly, here they have made, so filter papers are there, then your membrane is there, then gel is there, then more filter papers are there, like this you have to put. And then you put it for overnight, or like some for some hours. And then, okay, so I think we have completed. If you have any problem, we can discuss the Western blotting for a in the next class. If you want, if you have any problem, we can discuss it, because Western blotting is also another very important uh, technique. Okay, so anybody has any questions? Sorry. Yeah. Okay, then we'll wind up for today. And if you want, we can discuss the Western blotting in the subsequent classes, in the next class. And try to read about why blocking is. So, you know why blocking is, what is the meaning of blocking and why blocking is done?
Ma'am, to prevent non-specific interactions and subsequent false positive results.
Yes. So blocking is done to prevent the non-specific interactions, and blocking is done here also. We do blocking. Blocking is done with the help of BSA, bovine serum albumin, or you can have skimmed milk also. But highly sophisticated labs, they kind of use BSA also, and but milk works equally well. And when you're doing fluorescence microscopy also, like anywhere where you are having antigen antibody interaction, antigen antibody interaction always requires the blocking step. Why? Because you want specific interaction of your protein with your antibody. Your antibody can have non-specific interaction with 10 other molecules also, 10 other molecules also, but you don't want that. So you will block all other non-specific interactions, so that only your specific protein molecule, protein of interest, and your specific antibody gives your result. So whatever result you are getting, that you're getting only because of the interaction of the antigen, your protein with the antibody, and not because of the a non-specific interaction of antibody with any other molecule which is present. That is why blocking is very important. Okay. Okay, then we'll wind up here for the week, and then we'll meet in the next Tuesday. Now we'll meet on Tuesday only. Okay. Chalo. Bye bye.
Thank you.
Thank you.